Your red blood cells don't contain any DNA, they have no nucleus. We can now type people that are 6, 7, 8 generations away and make a relationship between the 5th cousin and the remains. She was adopted and she didn't know who her family members were. She used the test and she found her mother. There are people who have taken the test and they found out that their father is not their father. So you may present as a female, but you may not be that.
Is it even possible for a criminal not to leave any kind of DNA-related evidence?
They're looking in the air conditioning vents and they're finding human DNA in the air conditioning vents. DNA isn't the only thing that says this guy committed a crime or this person didn't commit a crime. It's part of the puzzle.
We have all seen a lot of criminal movies. So I'm wondering how realistic is forensic DNA analysis shown in those movies?
Well, that's a tough question to decide or describe how realistic forensic DNA is in the movies. Because in one aspect, forensic DNA is a very powerful tool that helps to identify people who have left biological evidence at crime scenes, like blood or saliva or hair at the scene. So that part is real. The part that is often not real is that in the movies, you have to do things very quickly because you have a short time frame to get the story. So normal DNA testing in a laboratory could take weeks before they actually provide a result. You could do it faster, but usually it takes a longer time. But in the movies, they put it in a machine and immediately you have all the answers. And it typically doesn't do it that way. There are machines that can speed up the process, like rapid DNA or other kinds of machines, but the routine work takes longer. Some of the things where they put the DNA in and you get the face of the person or that kind of thing, that's not really feasible. But the idea of having a woman who has been attacked and some DNA has been left on the woman, getting that DNA and acquiring it and generating a DNA profile that would be representative of the person who left the DNA behind, and then searching a database of people who have been convicted of crimes or arrested, that's real, and that search can happen in very real time. So again, there's some things that are realistic and some things that are more for the quick, short turnaround, because we don't have 10 weeks for a movie to occur. Yeah.
What cells can we use for this kind of DNA analysis? Can we use skin cells, hair cells?
Well, DNA in itself, when it first started, now DNA typing has been around for almost 40 years now. So it's now a sort of mature science. But before there was DNA, they used what we call protein markers, like the ABO blood group. You may be type A, someone's type O, and those were the only markers available at the time. They were not very practical markers though, because the ABO blood group, you might find it in blood, but you're not going to find it on a skin cell or a hair, something like that. DNA, not only was it powerful being able to tell the difference amongst individuals as who may be the source, it also had the power of it that DNA is in every tissue of the body. So if you leave a hair behind, I can take a swab of your mouth to get some DNA as a reference sample and compare it. If you left blood behind, or someone left saliva or semen or some tissue behind, all of those can be compared. And that's powerful because, if you arrest somebody, you're not going to get a semen sample from the person as an example or even a saliva. Maybe you have to do things. So if you can take any viable tissue and compare it to the evidence, that's very powerful. The other is for human remains. People who are migrating, these days we have lots of migrations because of war, poverty, crime, and so many other things that displace people. And people die along the way because of these events and the migrations. And you have these remains. So if you find remains, you don't have fingerprints, you don't have the eyes for eye color, you may not have the hair, you may only have some bones. And so you type the bone, you have some DNA. If you only find, say, some bones from the arm, then the other bones are somewhere else. But you can type the DNA from those bones and compare them back. And then also you can compare that DNA to a family member to try to look at relationships, because families share, some DNA. So you can use very different things. And I don't need the bone of the family member, because no one wants to give up their bone for typing, but I can get just a swab of their mouth and get the DNA I need to compare to the bone. So the real value of DNA is any tissue of the body. And we've typed in cases hair, skin, saliva, sweat, and any body fluid you can think of.
How distant can be the relatives so that to make this kind of comparison possible?
Well, this is a very interesting question, a very timely question. Traditionally, if we were going to do a family member, we needed what's called a first-degree relative. That's either a parent to a child or a sibling, a brother to a brother, or sister, brother, sister. Today, though, we have technology so powerful that is called whole genome sequencing, and we can now type people that are 6, 7, 8 generations away, fifth cousins, and make a relationship between the fifth cousin and the remains. And it's a whole technology called forensic genetic genealogy, where we do whole sequencing, not just some DNA markers, but your whole entire genome, your whole the 6 billion letters of your DNA code, and look for all the variants that occur, and then see how much is shared. And the more distant you are, the less you share. So you share half your DNA with your parents, and half your DNA with your sibling. But if you go to your grandparents, you share a quarter of your DNA. In each generation, you have that. So when you get to this fifth or sixth cousins, you may be sharing only 0.2% of the DNA, but it's enough when you have so much of the whole DNA to be able to make those associations. So if I make this association and this, I can then use some public records, look at their birth records, their housing records, and all these things to tie back to actually identify the person. So in today's world, we can look many generations or degrees away.
But don't I share the same 0.2% of DNA with other people that are totally unrelated to me?
Actually, you share probably about 0.1% of your DNA with human, well, you share 99.9% of your DNA with me, with anybody else in this room or anybody else, and that it's the 0.1% that we're looking at for the variation. So it's that 0.1%, which seems like it's very little, but that's 6 million places that we're looking at. 6 million is a lot of places when you think of genetics that you can compare. So if you have 1% of 6 million, you've got 60,000, right? And if you have 0.1%, you have 6,000. So you have many places in the genetics that you can look for sharing with that. So on a grand scale, it seems like a little, but on a grand scale, it's quite a lot of sites that we can look at to make those associations. And that's been a big technology for the past few years in the US for associating and identifying what we call cold cases. You have cases that go unsolved for many years. And now, because you can reach out to these more distant relatives, you can now find that distant relative who had nothing to do with the crime scene or didn't even know this relative, but you can trace back and identify some of these cases have been 30 years, 50 years, and unsolved.
I suppose the development of artificial intelligence is helping in these technologies a lot, right?
Yes, the artificial intelligence, machine learning, they're probably used interchangeably in this thing, is a real advance in science in general and lots of other areas as we've seen. But what it does is it takes what we do traditionally in science and throws it all away. So in science, we have to have a hypothesis where we say, I propose this, and then you attempt to disprove it, but most scientists end up trying to prove it. But traditionally, or purest way, you're supposed to disprove it. But hypothesis in itself biases you in the way you think, because you say, I think this is the way it should be. With machine learning, we don't have to know anything about it. We can just let the data tell us what's there. So you can say, I have this data, I want to find things, and the machine learning does it for you. The value of that is biology is a very complex field. And we as people are very complex. We as living organisms are complex. So we think because if we understand the fundamental DNA, we're going to understand what people look like and how they came to be. But it's more complicated because it's not just the DNA, it's the environment we live in, it's what we eat, how we do, what microbes we get exposed to in our life. All that shapes us and what we are. So it's such a complex challenge. Understanding the fundamentals in that has actually maybe derailed us from getting to what we need to do, is to address the problems that we want to solve. But with AI and machine learning, that helps us because we can skip over trying to understand it and find out what does it tell us so we can respond to it. And so that's going to be the future, starting now, but the future. And we'll see a lot of that in forensic DNA analysis and other forensic sciences as well.
I suppose that AI and machine learning also make the things faster, like the process of DNA analysis.
It could, and it may not. I mean, some of the DNA analysis that we've been using for many years, we don't need AI to do that. But what you can do is, if AI can approximate well to the best learned or experienced DNA analysts, like I've been doing it for many years. I helped develop it. I know it better than the younger person who's starting today. How do you transfer that knowledge base from this old timer to the young timer? There's always a gap in that process. If AI could approximate well what I know, and then have a tool that allows the younger one to do it, we don't lose that informational gap, that experience gap. And that could be an improvement. And not everybody does as well. There are some really good people, there are the majority in the middle, and then there are some that really struggle because they either can't conceive it well or whatever, but yet we want them all to do well. So if you have tools that can help substitute, not replace, but help in the process, to be a supporter in the way you're reasoning it and see how they match up, that can be a real value. Now, whether it's more speed or it's more credibility and confidence, I would prefer the credibility and confidence over the speed because that's really more fundamental. But if you could turn it around faster, that would also be better because it takes a lot of effort to manually review work. And if you had tools, and there are some that exist today, such as what's called probabilistic genotyping, which are software tools to help interpret DNA evidence. But if you had other tools that could go even further and know when to really interpret and when it's maybe we should step back, that could help a lot too. And I see more of that coming.
Getting back to the cells that can be analyzed, are there any cells or tissues in our bodies that cannot be analyzed this way?
This is a good question too, in that sense. Actually, in your blood, your red blood cells don't contain any DNA. They have no nucleus. So when you get a blood stain, all this, what you see, has no DNA in it. It's the white blood cells, the lymphocytes, that are in the blood that we're actually getting the DNA from. So even though the red blood cells don't have it, they're mixed with the white blood cells, and so you have DNA in those samples. The hair is an interesting one because we developed a different DNA test for that over almost 30 years ago now, called mitochondrial DNA, which is another type of DNA that you have in your cells. It's known as, the mitochondria are known as the powerhouse of the cell. That's how we generate energy, and then they have their own little DNA molecules in them. So you have a few hundred to 1,000 of these in your cell. So they're very good because there's many copies of them for detecting trace amounts of material. So for hairs, we couldn't get the traditional DNA because there's not a lot in there. There's not cellular DNA in the hair, it's just degraded. So we looked at the mitochondria as opposed to the more standard DNA markers that would be used, and that's been very successful. Even 1 or 2 centimeters of a hair shaft, we can get mitochondrial DNA profiles from. But now today with sequencing technology, we can go back to the hair because it does have the more traditional DNA in it, but it's so degraded, we couldn't analyze it. The fragments of DNA are too small. But now with sequencing technology, whole genome sequencing technology, we can sequence those very short fragments and get much more information out. So as technology evolves, more tools become available, more avenues come available to help solve crimes.
So if a criminal leaves some hair on the spot, he can be identified by that hair?
Well, again, identified is a loose term. So when we think of that is, when we have a DNA result from some evidence and we compare it to a person, that DNA result, first of all, is critical. You may get a lot of a result, you may get half of a result, a portion, or a very small amount. The less DNA you get, the less information you have. So I was thinking of you, if I only had your hair color, that would identify you to some degree, but not you alone. If I had your eye color, that reduces it to some people. If I then have the shape of your nose or your height or whatever, I can start to reduce it down more. Same with DNA. If I have a series of DNA markers, say 20 or 30, but I only get 5, some other people may have that. So I wouldn't say I'm identifying someone to this person and this person only. If I had more markers, it's likely this is the only person or an identical twin, but even identical twins aren't exactly genetically alike. So with technology today, we can even differentiate twins. But the idea is, the less I have, the more people can be included. So the question could be is, if you're arrested for the crime and I have DNA evidence I found at this crime scene, I type it and I ask the question, can you be included as a contributor or excluded as a contributor? If you have the same DNA profile, you're included. But if I have limited information, so maybe another 1 out of every 1,000 people. So it may not be identifying you, but it does have value in the courtroom if they have that, plus they have your face on a video camera, they found your fingerprint at the crime scene. So the other circumstantial evidence combined can be very powerful. So you don't have to have a unique identification to add value, but you can get closer and closer with more and more genetic information.
What about the skin cells that we shed?
Well, there's a whole field of, subfield of samples in DNA called touch DNA. It's probably not a great term, but it's the term that's used. So when you touch something, you leave some of your DNA behind, and there's some belief that some of these shed skin cells. I'm not convinced they're all shed skin cells, because when you talk to people, you watch people during the day, what do you think people do during the day? They go like this, they put the pen in their mouth, they do this. So you get a lot of DNA from your sweat, saliva, mucus, and things that are on your hands. And when you transfer those, you tend to have a lot more DNA. But people shed their skin cells, and they have DNA in it.
Those are supposed to be like dead cells?
They're dead cells, and that's another limitation. Because as these cells die, in a term that's called apoptosis, they start to lose their nuclear DNA, the DNA that we're focusing on. And so they produce less DNA than if you had a fresh set of viable cells. But they do have collectively DNA in them, and so it depends on how long you handled it. If, like if you own a gun, you're playing with it every day, you're going to leave a lot of your DNA on there because you repeatedly handled the gun. If I handed you my gun and you shot and killed this person to the right of us, who's going to be convicted based on the DNA? Probably me, because my DNA would be there even more so than yours, because I deposit a lot of DNA. Now, I might not be convicted because there's an expectation if I own the gun, my DNA should be there. So it gets complicated sometimes, because if you live in a house and let's say your husband decides to kill you, or better yet, you decide to kill your husband because you would prefer it that way, and we find your DNA, a blood spot on the carpet, so a small spot. How do we know that has anything to do with the crime? Because you live in the house. So DNA that's there for reasons that may have nothing to do with the crime scene complicate when you're doing cases where you can find that. So if it's my gun, my DNA is expected to be there, but it wouldn't prove anything about me handling it when I fired the gun. On the other hand, if it's my gun and your DNA is on it, that might be important if you had no access to the gun, but you came in and stole it from the house or whatever it may be. So under the circumstances, it could have value or it could have absolutely no value. But those are, again, those skin cells, or maybe some saliva that you got onto your hands that we call touch DNA. and many items. And that's a lot of work these days in the crime labs, is being focused on this touched DNA.
I wonder, how do experts deal with these kind of situations, really?
It's a complicated one because there's a lot of issues. As you get to less and less DNA, what's called the touched DNA, much of it falls. Sometimes you get a lot of DNA, that's one thing. But when you get less and less, there can be many explanations for how the DNA got there. Now, when we type, we can't tell you how the DNA got there. We can only tell you there's DNA, and maybe you're the source, or maybe this person over here is the source, and this person's source. But when it was placed there, or how it got there, you cannot determine that by DNA alone. You have to look at other kinds of information. So when you get to very small amounts of DNA, that's at trace levels. If I shake your hand like we did when we first met, some of your DNA is on the hand. If I now touch an item like a knife, some of your DNA may be transferred to that at these trace levels. Now, if I touch it, you're not going to get large amounts of DNA, but there'll be some DNA. So now, a trace level of DNA found on a knife may be there because you were involved in the crime, or it may be because there's some secondary transfer process that brought it there. So as we get more and more sensitive, and we have more of these issues, it complicates the meaning of the evidence. It doesn't change the fact that you found DNA that matches or cannot exclude a person, but how it got there may be more than one plausible explanation. And that's something you just have to be honest about. You can't say, I work for the police and therefore it must be a crime that's committed. We as scientists have to say, that's one possible explanation, this is possible, and it's up to the court to decide on whether that's meaningful or not, based on the totality of the evidence.
Is it even possible for a criminal not to leave any kind of DNA-related evidence?
Is it possible for a criminal? I think it's possible, and in fact there are cases where you don't find any DNA or non-detectable levels of DNA, and that's so it's possible, and there are people who have tried to thwart it, and some have been, I think, more successful than others. There's a very famous case of a woman who was abducted and raped, and the perpetrator was eventually caught, but he had shaved his body and everything, and he repeatedly, he abducted her and repeatedly raped her over a series of weeks and that. And unfortunately in that thing is they found no hair evidence, and he cleaned her up and all these things. I think he used condoms and everything. And, but they found one small dot of blood. So somewhere along the way in the van where she was held, that one small dot of blood was all the biological evidence they found. And that actually was the one that linked him back to him through the DNA and the database searching approach that was used. So he tried very well, and he almost succeeded in that sense, but that one small dot there. So depends on the type of crime. I mean, the more violent it is, I think it's harder, but the less violent it is, it may be easier, because you can put on a mask and gloves and things, and you might not leave any traces behind. So I think it all depends, but so far it seems like they like to leave DNA behind, or they don't understand it well enough to worry about it.
In such cases, do experts have to check everything in that territory to look for DNA traces, like everything?
Well, it's not practical to type everything, because that's a lot of work, and the labs have a lot of cases. Now, I believe the police should collect everything, but I don't think you need to type everything. Now you have to use your intelligence and understanding of the crime scene to focus on what may be the most meaningful set of samples, the most probative samples to type. Because if the police came in and said, I collected 500 samples, type them all, you could ruin your budget for the entire year. You would type many samples that have no value in that. So you have to be intelligent and make choices. Start with maybe the first 5 or 6 samples that are meaningful.
But how do you decide that?
You have to know this is where the crime scene comes important. So the crime scene investigators, when they go to a crime scene, they're going to secure it, they're going to document it, and they're going to try to reconstruct the crime scene based on how they collect the evidence. So if you have a rape case, the first and best sample is the swab taken from the victim, right? Because you know where that happens. And then it might be the bed sheets. You might think the doorknob come in, but if what the person never If a person knocked on the door and the woman opened it and he pushed the door in, the doorknob becomes less valuable. So there's more uncertainty about a doorknob or the wood frame on the door or something. But there's more certainty about maybe skin cells on the linens on the bed, or swabbing the victim where he may have licked her or something like that, or the vaginal swab from a rape. So you focus on more likely places first. Now, if it's a really serious crime, you want to solve that, like a mass murder or something like that. So you're going to try to identify them, but you can't type everything. So you start with the top samples, and then if those don't work, maybe you go to another set. But after a while, if you're doing more and more, you're probably not going to get there, because if your intelligence says these aren't working, then the more you look, you're probably not going to find something. And then there's DNA everywhere. So collecting DNA and getting a profile doesn't necessarily mean it had something to do with the crime. So you don't want to collect everything, because you'll get a lot of information, but it will have no value. So it's really important to focus based on the crime and what are at least the indications of what may have happened in the crime.
Have you personally worked with that kind of difficult cases?
Oh yes, I've been involved in many difficult cases, only because nature is, I was the one, I worked at the FBI. I was the one who was the lead to develop the DNA program for the FBI, bring it onto operation, and help build the database. So whenever in our legal system, which is much more challenging than your legal system when it deals with scientific evidence, I'm also the one that had to go in and defend it in the courtroom for its legitimate use. And so involved in lots of different cases. And then after I left the FBI, I ran a lab in Texas, and we had lots of cases. And there are many different crimes of nature that are, there was a case I remember early on where it was a man wanted to date this woman. She had a 2-year-old child. She went out once. She didn't want to go out with him again. He wants to go out again. So he's knocking the door, he breaks in the door, and he stabs her 30 times, child 20 times, and they're dead. And as he stabs, he leaves some blood behind because he cuts himself. So you have to go through all this blood and find the blood that's his, and those kind of cases. We had a case where the Hells Angels, you've heard of Hells Angels, and that to join the Hells Angels, you go through initiation of killing a member in a rival gang. So they have this whole watcher, driver, shooter, and they went to shoot the person they thought was the member of the rival gang, and it was just a person who had a similar van. So they killed an innocent person, or they killed a person regardless, and the bullet ricocheted off of the van after it shot him and hit the shooter in the arm and left the blood, and then he left, and we had to find him later through lots of different means. So you have those, and then we had one case where, I'll just give one more, because it was a hair case, and you were interested in hair, was a man who had an 8-month pregnant wife, and it looks like he killed her, dumped her body in the San Francisco Bay, and there was a hair in a pair of pliers, pinched in the pliers, that became the critical evidence. And so we used that mitochondrial DNA, and it matched the dead wife's hairs, her mitochondrial DNA, and that was part of the evidence to help convict the person. So that's very trace level material and very unique circumstances. But that was a big media one where all the news was there. You had to walk through crowds of news people to just even get into the courtroom. And why that one as opposed to another one, we never know. But so these are things we have to deal with regularly.
Was the husband the murderer in that
case?
Well,
I'll say this much, he was convicted of killing her. And the evidence was, I think, convincing, but that's a decision for the juries. Whether he was or not, only he knows for the truth. But, all the other evidence, which I don't have time to go into, it's strong evidence that he murdered his wife, yeah.
Which was the most difficult case to solve?
Oh, and people ask me this, and I really don't have a way to say that, because all these cases are important, and all of them in some way are difficult, because we're just, DNA isn't the only thing that says this guy committed a crime or this person didn't commit a crime. It's part of the puzzle. So we're just a part of it with all the other ones. Sometimes it's stronger evidence, sometimes it's corroborative evidence, but it's all part of it. But to me, everybody asks me, what's your most difficult case? Which one, ones you remember the most? It's really hard to say because they're all important cases. And they're all very, I don't have a case I remember where it's just like someone breaking and entering into a house. It's always been a rape, a murder, kidnapping, or something of that nature. So to me, they're all difficult in some way. They may not be technically difficult to generate a DNA profile, but ensuring that you have good quality, that you've got the proper paperwork, that you're ready for going to the courtroom, that's all take challenge, and that's not an easy thing for people to do. And you've had a lot of experience, you're more comfortable, but for some people it's very hard. But I wouldn't want to put a difficulty, because we have to always have the same high quality level when we're doing our work. So I'd probably put it more like that. But some cases are harder because they have less DNA, or they have, complex situation, or, it's a missing persons case where you find some bones, and now how do we match it up? Or there's been an airplane crash, and you have a mass disaster. Now you have all these bones and you have small pieces, and you have to match them up to families. Those are very challenging because you may have 200 people die, but 20,000 pieces of bone. And if you want to identify every piece to a person, that's a lot of work. That's very challenging. And not all pieces have good DNA in them. And then sometimes you have a situation where family members died and you have two brothers, for instance. DNA typing can't tell us the difference between one brother or the other by looking at their parents. You only know that they're brothers. You would need to have some, what we call an ante-mortem sample, such as they licked an envelope or they have a baby tooth that you can say came from this one brother, not the other, which is also challenging because parents don't write, this is this person's baby tooth, they just have the baby tooth. So these are all very difficult, and each case has its own nuances that we have to consider.
Is it challenging to work with half-eaten foods? Can we take DNA samples from half-eaten, burger or something?
There have been a number of cases that have been solved, or at least helped to solve from that. In fact, there's a very interesting case. Let's take it a slightly different way. So there was what's called the Grim Sleeper cases when it was out of the Los Angeles area, where a number of women were raped and murdered over a number of years, and they never could solve it. And the DNA didn't find anybody in the database to solve. And they did what's called familial searching, and this person's son was put into the database for a weapons charge. And so when they searched the database for a family member, just one generation, 1 degree away, they got a hit. So now the thing is, well, how do we now get a sample from that person they got the hit, who's not in the database? So what they did is, they had some police officers dress up in waiters' costumes at a pizza place, and he went in to eat with his family, and he had some pizza with the crust on it, left it there, so half eaten, and swabbed it, and got a DNA profile that matched the evidence, and then they could get a court order to get a reference sample from him, because in our country, once you throw it away, it's fair game for them to analyze. So there's a half-eaten example, but it's not the evidence in the crime. But I know there are cases where they found food half-eaten at a crime, and they can match up to DNA. Because when you bite, you do leave some good saliva behind. The other is drinking cups and beer cans and things. You find people who break into houses, they like to go in the refrigerator and they drink something, they throw the bottle down or something, and usually swab those, you get a lot of times very good amounts of DNA.
It's strange that some criminals are that kind of careless. If we can say so.
I don't know if it's careless or,
or do they really want to be found? Because there is such a theory.
Well, I'm not sure that a person who commits crimes wants to be found in that. I mean, the idea of committing a crime is so you can do whatever you're doing in that, but if you get caught, then you're not, well, maybe you get free meals afterward and you get free housing, but no, joke. But no, I think a part of it is people don't really understand the DNA, and that they may watch things on TV, but it doesn't necessarily connect. to the power of it. There are some people I think know it and they try to subvert it in some way or try to circumvent the process. But I think a lot of people just don't realize it, even at this day, that not just the criminals, but I think law enforcement itself isn't as educated as they should be either. And we've had situations where the police officers have been contaminating the evidence unknowingly, and that because they're not trained. So what you may know because you're interested in the topic area, or we may know because we do it, may not translate well to the general public. And even what you see on TV, even though you hear it, you may still think, well, it's TV, it's movies. I mean, they have dinosaurs on TV, but we know they're not dinosaurs. So maybe they don't really connect with what they see on TV as being real. And so maybe there's a big gap in education and training.
Sometimes it's for the best, I guess.
Well, hopefully it's for the best that they don't learn about it. But even many criminals who try to circumvent, they tend to still leave something behind. So we're still better off. But if you really wanted to do that with DNA, there are ways to reduce the chance of leaving DNA behind. And so the less they understand, the better off we would be. But we live in a world with information at our fingertips. So who knows what the future may be. Yeah.
Are there any cases where it's not possible to identify the body or the bones, for example? Maybe if they are, I don't know, burned or something?
Well, burn's a good example of that. Burned bones, after, depending on how much they've been burned, will come to a point where you're not going to get DNA results. So usually when we look at these cremations as an example of the extreme end of the burned bone, you often can't type those samples. There are some where burns are not so severe, so you can see some discoloration on the bones, but in the bone there's still good DNA. And there are others where there's no detectable DNA. Sometimes you get a sample that's not burned or anything, but it's been in the environment for a long time and exposed to the sunlight and microbes or minerals in the soil, and those sometimes yield no DNA. And then there are other times you had a bone that's been sitting for 20 or 30 years. In fact, we did a study on a bone that was 170 years old, and we got a full DNA profile from that. It was just really good DNA. So sometimes just the luck of the draw. And then the Nobel Prize this past year was on work by Svante Pabo on work, basically generating data for the Neanderthal. So you're talking about 40, 50,000 year old samples that have traces of DNA in them. Now, not every one of them obviously does that, but some do. So what makes one sample successful and another one completely gone? That's still some things we don't quite understand. We know there are things that happen and what are the environment that affects that. But in any one bone, you can't predict. So I can take a fresh bone and get a full profile. Another case, get almost nothing. I can get another one and it's really old and get a full profile or very little. We were involved in helping analyze some bones in Cyprus after the war between the Turks and the Cypriots. And so these were from the 1960s, and the military had maintained them, but they sprayed them with something, some chemical, on a regular basis. So these bones were blanched white and that, but when we extracted them, they started fizzing up with some sort of chemical thing we didn't understand. We could get no DNA out of those, but yet they weren't buried or anything, but they were chemically treated in such a way that we don't know what the chemicals were, but we could not recover anything. So you never know. It's circumstances of each case.
Now I wonder why they were doing it.
Again, why they did it, I don't know. You can speculate, but without knowing. They may have been spraying them because they thought they're biological material and they're preserving them. Who knows? Or maybe this was a sanitation thing because human remains you need to clean because you get disease from decaying bodies. These were bones, so I don't know why. But whatever they did really made it impossible to type those bone samples. And my colleagues there spent many years trying to resolve that with very little success because it's just the circumstances of those set of samples.
How small can a sample be for DNA analysis to be even possible?
Well, that's the thing is, every day, every new technology gets smaller and smaller, but in the equivalent of a handful of cells, a very few, 5 to 10 cells, we can now get a tremendous amount of DNA results, such that in some cases even a full DNA profile, and with this sequencing technology, many of these genetic markers. So in one case, you may not even, like the touch DNA you were talking about, you may not be able to see the DNA because it's invisible in swab. So that might be 10 cells or 20 cells, equivalent of DNA, but you can get a lot of results from that. But when you get down to 2 or 1 or a few cells, it becomes more difficult. But in theory, you can type a single cell and you'll get some information. But then the question is, what does it mean when you have a single cell? Because that could come from anywhere. It can come from anything. So even though you may even have the capability to get some results, then you have to say, well, what does this mean? So at some point, being that exquisite in sensitivity may have diminishing returns. But in theory, we can type single cells now, but we don't get a complete profile of a single cell because there's some hit or miss in that. But you can get partial information.
Yeah, if I'm not mistaken, after the Second Artsakh War, there are still some bodies that have not been identified. So what could be the problem? Are there any technical problems that make it difficult to identify them, or?
So generally, those samples tend to be better quality samples because they're more recent, but sometimes they can be very degraded. So you have the problem of just, are there sufficient DNA in the bones? And you won't know till you find them and you extract the DNA. The second is, when you have a bone now, how do you identify the person? We now have to have either some item from the person or a family member. Most of this work is done by having family members. So now you have to have family members provide samples. Some family members are very eager to do it. Some family members don't want to do it. Some trust the government, some don't trust the government. And you have all these things. Well, some just say, it's done, I do not want to be involved anymore. We know what happened, so that's it. Others want to find their remains. They want to give the dignity and respect back to their missing person and bury them and have some resolution. So it's a wide range. And that's where the real challenge is to get those family members to be able to identify. So with the standard markers, we're looking again for a parent or child or brother or sister. If we now do the sequencing of the whole genome, we might be able to find some relatives more distant. You may not know that, but your third cousin may be these remains here. You would never know who that person is. Likely don't know who your third cousins are, but now there could be a link to solve. But that finding those family relationships to identify the person is going to be one of the bigger challenges.
Are you familiar with how is this all done in Armenia, DNA analysis?
I'm familiar with some because we just spent the last few days with them, and I have to say, they're building their programs. You have some established lab and you have some that are growing, but they seem very bright, very capable. They understand the issues and the problems. They understand what they need to do, and now what they're trying to do is build their capacity to be able to do more things at a higher volume. But they're running cases now. And from what we discussed with them this week, they seem to be very knowledgeable, and I think they have the talent and capabilities. But over time, where we've run hundreds of thousands and thousands of samples, they've run fewer, so they won't have the same level of experience. But from what I understand and what I've seen, I think they've got a lot of experience, and they're moving in the right direction to be able to provide more things over time for Armenia. So I think you're in good hands. And I think the way the system is developing here, with the structure and the way you have this new, investigative committee, and you have the Bureau of Expertise doing work, and you have legal medicine doing work, that you seem to have all the things in place to really do a good job in this area. So we should have high expectations that they'll be your next CSI news story that you'll be having on a routine basis.
We can say that DNA analysis has revolutionized forensic investigations. Do you see any kind of ethical problems that it has caused?
Well, there are ethical problems in anything we do when we collect information from people. The standard DNA markers have little information to tell me anything about your health, whether you're going to be a high risk for cancer or diabetes or things like that. But that whole genome sequencing, that gives me all your DNA information. So there are some risks, and so we have to have proper security, privacy, maybe data encryption to do that. So that would be something. Some countries like the US, when they collect the DNA sample from the person who's convicted of a crime or arrested, they keep the sample. So that's a risk, because if you have the DNA sample, even though you didn't type it for those personal, private genetic information, it's accessible. Some countries, they take the sample, they type for the standard forensic purpose, and then they destroy the DNA sample. So there's no risk of someone getting at that sample and being able to type it. So these are things you want to think about as you develop to, and everything is a risk-based assessment is, what is the possible risk? Because there's always risks. You know, there could be a nuclear explosion today, you could destroy all the evidence in the labs and all the evidence in the city and everything. But we look at that, even in the world we live today, as a low risk, high impact, but low risk. And that there are other things like you could have a severe storm that, you know, like a tornado kind of effect that destroys the lab. That's probably a higher risk, but maybe not enough to destroy the lab if it has things in secure lockers and things. So we weigh out all these risks and see, is it a high risk of high impact or of low impact, low risk of high impact and low impact, and make decisions on how we want to safely and securely protect our information and the people who may be associated with that. So there are always some risks, and there are others where it may not be a problem. So if I had your basic genetic profile that's used in forensics routinely, that the labs here use in this country, I couldn't really tell you anything about you. So if it was disclosed, not that I'm advocating that, but if it was disclosed, there's really low risk to you on a privacy level, but we still don't want to disclose it because we want to protect everything because of the purpose how it's being used. But the risk of something affecting you privately is very low. The risk of someone accessing it might be higher because someone in the lab might be a terrible person and break all the codes of conduct that you do, which I don't think would happen with the people. But if it did, that's a higher risk than your privacy. But, so we have to weigh that out. We as forensic scientists believe in integrity and trust. So the honesty is really important. So it's rare, although it's happened, that you find people who are contaminating evidence and doing these sort of things. So we would like that as a low risk, but it's a high impact risk. So you have to decide where those are, and that's up to all the people involved to determine when that would be. I'm not so worried about the basic DNA profile. I'm more worried about the DNA itself. So you would have discussions and decide where that may be.
Many people today take those popular genetic tests to trace their ancestry. Yeah. This means that some third-party companies are getting like a lot of personal information. Does these contain any kinds of risks?
Well, you know, it's interesting you say that because everybody who does that test, there's this terms of agreement and contract that you're signing, and most of the people don't read it. That's the problem. And in some of those, they have taken their samples and used them, and some of the direct-to-consumer companies have actually sold those DNA data and made hundreds of millions of dollars off of them. So I don't know if everybody really anticipated that you gave your DNA up and now it's being used by someone else to make a profit. So there can be some issues in there, but they do sign those terms of agreement, and if you don't read the details of it, you know, and even if you did read it, you might say, oh, so what? It's just another DNA profile in that process. Now, I have never done it in that. I've never given my DNA to the companies because I have no desire to know. Because, you know, there's an old joke about it. If you really want to find your third cousin or your distant relatives, you could do this DNA test, or you can just put in the news media that you won the lottery, and you'll find a lot of relatives.
That's true.
Yeah, right? So there's different ways to do it. Yeah.
Who could be interested in purchasing that kind of data?
I can tell you there's a lot of people that are interested. In fact, the technical leader in my lab when I was in Texas, she was adopted and she didn't know who her family members were. She used the test and she found her mother. Her mother was in one of the databases. And so she went and met her mother. And now they have some relationship that 50 years later. So there are people that find their long-lost relatives, so that's there. There's also the other side of the coin where there are people who have taken the test and they found out that their father is not their father. And sometimes it's because the mother, and whatever happened. And others, there's these fertility clinics and you thought it was the father's sperm that was donated, but it was the fertility doctor was donating his own sperm. And so they had lots of people who were related in the community, but they weren't the parents of that. So it's destroyed relationships because, you know, you think this is my child and it's not. And it's brought the relationships together. So information can be great and it can also be damaging. So you have to also know, again, the risks whenever you take on more information and that. So there can be good consequences out of them and there can be bad consequences. And it's not for me to decide whether I should tell you or not. It's up to you to decide what information you think you should look for. But you should know the risk before you start. And I don't think most people know the risk.
Who would be interested to buy that information, the information about the clients from the companies that perform those DNA tests?
If you have information about the whole of DNA, there's hidden information in there that can be quite useful for developing pharmaceuticals or therapeutics to prevent disease. So take like HIV, which was a very terrible infectious disease, right? And anybody who's exposed seems to be they're going to die from it if there's not treatment. But there were a small group of people who were exposed and never got infected because they had a genetic variant that didn't allow the virus to integrate into the cell, invade the cell. That genetic information is a clue that might be able to develop a drug. And that company who owns that could make billions of dollars if they could develop into a drug. So all of us have a lot of genetic variation, and we probably all have something that could prevent some disease or make us susceptible to another disease. And so being able to mine all that information and be able to exploit it is very powerful. So spending $200 million to get all that DNA, where I might end up developing a drug I can sell for $10 billion, is a small investment. So, but we don't know what the hidden variants are because there are people who are exposed to drugs that they will die if they take the drug, and others who may not die from it because of their genetic makeup. So knowing that also is of value because you could develop a diagnostic test to say, this person shouldn't take the drug, this one should take the drug, because we want them to have benefit from it, not to have something that's going to be, you know, kill them or something like that, some adverse drug reaction. So all this knowledge plays into that. And so the more DNA I get, the more variation I have that might someday give me that drug. So from a drug company, very powerful.
In what other spheres can this DNA analysis be used other than forensics, these ancestry tests?
Well, it's always interesting because you never think about some things until you get into a situation. So I have a colleague in South Africa, her name's Vanessa Lynch, and she's a lawyer actually, but an advocate of DNA. So she knows more DNA than the average lawyer or something on the street. But she then went on and did her doctorate in gender-based issues. So we think about gender, but in DNA typing, we don't type gender, because gender is a choice. We type biological sex. So whatever is on your chromosomes, you're X and Y, you're a male, X and X, you're a female. And they report that because they don't see that as personal information, because I see you as a female, right? But that may not be your true biological sex. You may present as a female, but you may not be that. So when we present this evidence in the courtroom, it becomes public information. And that public information is saying this person is a male, but may present looking like you, saying they're a female. And if it doesn't really have an impact on the case, we've now gotten into some privacy issues on the individual. And we've never really thought about this before, but she saw this thing, and we just wrote a paper on this together in this area. And so that's an example of a privacy. The other one is, which I wrote with a colleague of mine out of Finland, his name's Antti Saljantala, who he's a pathologist, so he does autopsies and that. And we always think about the genetic data. When you have someone who dies unexpectedly, whatever, there's an autopsy done to try to determine cause and manner of death. And sometimes from the information, one cannot tell what was the reason for that. So sometimes it whittles down, you whittle it down, you whittle it down, and then you might have to use genetic information to determine what might have been the cause of death. So sometimes you have, so an example was, there was this 13-day-old infant who died of morphine poisoning. So you know that the infant wasn't going out and getting drugs because he's 13 days old. So that now looks to be an infanticide. Maybe it was the parent or a boyfriend, or maybe it was negligence in the pharmacy. They gave the wrong drug. But what it was is the woman was given an analgesic because she had an episiotomy for pain. And of course, those analgesics, you metabolize them into morphine. That's how you get your release of pain, okay? Or now you're inert to pain. But she had a genetic makeup where she's a rapid metabolizer of opiates. So because she was a rapid metabolizer, she had high levels of morphine in her blood. She was breastfeeding toxic levels to her child. So genetic information can help determine cause, or in this case, cause and manner of death. So this is an unfortunate accident that occurred because of not knowing the genetic makeup. There are other people who are like, you see the story where you have that athlete that's really fit, and you see them jogging and they die. But so you don't have a lot of medical records on young people and athletes in that. But there are certain genetic disorders that affect the way your heart contracts, and under stress may cause it to pause for a while, and you end up having a heart attack and die in that. So now you have an autopsy, you don't have any bad heart conditions, but you have this genetic makeup that tells you something about the way your metabolism works. And so using the genetic information, you can then determine what might have been the cause of death. The problem with that is that genetic information is in the report, and it may be described in the courtroom. And now it becomes a public record. So that genetic information is also available to the public and to the family members. And there's no genetic counseling to help the family members, or now potential privacy to the family members who may be at risk. So there should be some other mechanisms in place to at least counsel the members, or do you need to put all the information into the court record, or just keep it in the case file and you talk more general about it? That's a hard issue in the legal system, but these are other issues we have to think about as we become more and more reliant on genetic information.
How do you see the future of forensic DNA analysis? How is it going to develop? I guess the process is going to become faster, but what else?
I think the best way is, I see the future is bright.
That's it.
No, I think, it's a very well-established technology. So what we do today, I see that going on for a number of years. I see the sequencing technology advancing, getting better. When the human genome did the first sequence, that cost $3 billion and took a decade. So that was not something that was going to transfer into the routine work. Today, we can use this whole genome sequencing technology, and for anywhere from $600 to $2,000, sequence your entire genome. There are now technologies that are saying they can do it for $100 or $200. So over time, we're going to see advances that are going to make it faster, cheaper, better. And I see that happening over 5, 10, 20 years, whatever it may be, that the technology keeps advancing. And those tools will be moving forward. But at the same time, we're going to have to think more and more about how to encrypt the data, protect the data, and make sure it's only used for the proper purposes.
Do you believe that one day DNA analysis can be used to predict a person's behavior? Like, can he be violent in the future? Is he a potential criminal?
I think it's a funny story because I've actually been involved in a case on this already. There is, and there's a case in Italy where because of the genetic profile of the person for the specific gene, the judge reduced his sentence by a year, didn't complete way, because of this genetic predisposition. But there is a gene known as the warrior gene. You can look it up. It's called monoamine oxidase. And there appears to be some association with the genetic type and being of this aggressive criminal behavior. And the problem is, not everybody who has the type has that, but it seems to be related to people who have really abusive or traumatic childhood. So if you're an abusive family or these things, or you were, beaten and all these other things, or parents were drugs and you were exposed to drugs or things, this category of individuals seem to have a higher propensity for this aggressive criminal behavior. But it may be true. People with more aggressive and successful behavior, maybe, but the environment they grew up in may not put them into this category. So there's some evidence for this, and it's gone into some courts, but the courts are reluctant to rely on some genes to predict behavior because there's so many other factors. So genetics alone doesn't predict behavior. You need to have other factors. And we are at some point both supposed to be responsible for our behaviors. So how that plays out, I don't know. But there are some already of this. So you should look up the warrior gene.
So we have something like a tradition in this program. I tell a famous AI about the guest I'm going to have and ask if there are any questions you would like to ask. If DNA could talk, what secrets do you think it would spill about us? Well, I guess DNA can talk in some sense, but we do not really understand its language well yet.
Remember, DNA is talking to us by what we do, so in a sense it does. It doesn't just tell us all these things we talked about, but I can tell you by looking at DNA what is your ancestral heritage, because your DNA has signatures going back as far back as humans around. So, my ethnic background is different than his ethnic background or his ethnic background, your ethnic background. And with enough information, we can really hone that down quite a lot. So it tells us that. DNA talks and tells us what we may be at risk for. You think about it, you would know it anyway, but a person who has sickle cell anemia, that's one little change in those 3 billion or 6 billion letters that changed. And that's telling us something. Now, we would be able to tell because we could see the person manifesting disease, but we could also tell genetically as well. The other might say is, I have all this DNA. Why are you treating the body that I built so badly? Why are you eating these bad foods? Why are you not exercising well? And my DNA, even if I tell you you have these problems, still eating healthy and exercising and getting good sleep is better. And why are you not taking care of this body that I built? Who knows what they may say.
What would be the most unexpected item to contain valuable DNA evidence in a crime scene?
The most unexpected item?
Yes.
Gee, I really don't have an idea for that, because just about anything that comes from the human body, we expect to have DNA. Maybe I'll put it a different way. There's now what's called eDNA. There are people looking at environmental DNA. So typically we have to think about someone touching an item, or if you talked and saliva came out. But there was just a study where they're looking in the air conditioning vents, and they're finding human DNA in the air conditioning vents. So you wouldn't be thinking of looking to solve a crime there. And they're saying people who have been in the room for a long time periods tend to be inside the vent, and those who are recent tend to be on the outside. I'm a little bit suspicious. This doesn't make a lot of sense, because how does recent DNA know to be on the outside and not be on the inside? Because you would think that. But there's some suggestion of places that we wouldn't normally look may have trace levels, which is interesting, but also says that's meaningless information, because if it's being collected all the time, what am I going to do with that if I get a result? So it may be there, and if it's filtering through the air, who knows what's floating in the air, a single cell floating and getting trapped on something. So having the information is interesting. not where I expect to look for DNA, but intuitively it makes sense if we're all shedding DNA all the time. But again, what am I going to do with it?
Which historical mystery would you like to try to solve using DNA analysis?
It's funny because we were just talking about this today. I don't believe we should do any of those historical mysteries, because if you type the sample and it does not belong to the person you believe it is, you have destroyed all your beliefs, your myths, your legends. So I worked on a case where it was a saint from the 13th century out of Sweden at the lab, and they had these two skulls. They believed it was the saint and her daughter. And so if we typed them with certain DNA, they should match up, the mitochondrial DNA. They had different mitochondrial types. So now we have a problem. One skull could be right, one could be wrong, this one could be right, this one wrong, or they could both be wrong. And so now you have a problem of the credibility of what you believed was this saint's bones. And that, so you run a risk. So sometimes it's better just to believe and have faith and have the mysticism of it, and other times it's not. So I'm more of along the lines as, if I had some ancient remains and I believed it was that, I just want to believe it. Because if I type it, the odds are probably likely it's not. And so these can be the issues. But there's been some interesting cases of that. George Washington's family was just reported. There's remains of Beethoven's hairs, Napoleon's hairs, Lincoln, Abraham Lincoln and that. So it's interesting, it's nice academic, historical, but in some cases, maybe it's better just to believe.